Good evening, all. Uh, I'm Dr. Sen from the Department of Genetics, Immunology, Biochemistry, and Nutrition. And I have the proud privilege of introducing Dr. Prakash Gambhir, who's our guest speaker for the series on genetics. And as you can see, Sir has put the introductory slide where the topic has been mentioned is for today is how to investigate a genetics patient, and it's basically which, why, and where. And a quick recap on Dr. Gambhir. Sir has got a long biodata, but I'll try to bring out some salient points. So, sir is a a pediatrician who has graduated way back in 1986. He's finished his MD by 86, and sir that year topped in the university and got the best award from Savitribai Phule Pune University uh, departmental award. He has done his fellowship in Germany in 1991-92, where he was involved in training in enzyme kinetics, Western blottings, etc., in the genetics lab and genetic counseling. Sir has almost 15 years of experience since 1985 in BJ Medical College as a research officer in the Department of Genetics, in the field of genetics. And he was one of the first ones to be part of these 5-center conglomerate, which was looking into this ICMR multicentric task force project in human genetics, along with All India Institute, Delhi, SGPGI, KEM Mumbai, and St. John's Hospital, Bangalore. Sir has almost 18 years of experience in BJ Medical College in the Department of Pediatrics as a assistant professor. And thereafter, Sir was a professor simultaneously sometime from 2007 till 12 as the professor head of this very department which he established, that is the departments of genetics, immunology, biochemistry, and nutrition of MUHS. And he was the first one involved in setting up the molecular workstation with various molecular, techniques such as real-time PCR, and was also he played a key role in establishing and starting curricular courses like medical genetics and reproductive genetics. You all saw that presently Sir is with Life Cell Diagnostics, and he's also the chairman of Genetics Chapter, Indian Academy of Pediatrics. Sir has got multiple publications, and also he has many presentations in India and abroad. And Sir has been involved as a visiting faculty for various different universities apart from MUHS, to name a few like Jivaji University, Gwalior, Gujarat University in Ahmedabad, and Institute of Human Genetics in Ahmedabad. Sir is a reviewer of multiple journals nationally and internationally, and he's got a vast experience in the field of genetics as the past Vice President of Indian Society of Genetic Screening, the Secretary of Indian Academy of Pediatrics, etc. And today we are here to listen to Sir about his experience in the field of genetics as to how to approach a patient with such parameters. Over to you, Sir.
Thank you, Dr. Sen, sir, and for this very kind introduction. You said many words. I hope my lecture stands the scrutiny of you and people. So I'll begin my lecture. I also thank Dr. Manasi Kunte, as well as our Honorable VC, for giving me this opportunity to present this lecture. And basically, it's a topic which is very vast, but it is very required for all the doctors in present generation, practicing as well as who are teaching in colleges, not just for the, discipline of pediatrics, who usually deal with rare diseases and genetics patients, but also gynecology and obstetrics, medicine, oncology, maybe other subjects, even like infectious diseases like microbiology, because genetics and the genetic techniques are coming in a big way. So every day, sometime or other, you may have to think of techniques in genetics. So I'll begin with the caveat that though this lecture was initially planned for pediatricians, still I'll make the things easy and as broadly applicable to other disciplines also, not just allopathy. Maybe if there are students or teachers from disciplines of, say, Ayurveda and all that, they will also find this easy to understand as well as easy to bring these techniques into their practice. So I'll just begin by what genetics techniques don't do. And this is because there are so many techniques that are there and everybody knows about Human Genome Project and genome sequencing and other things. And then a simpleton solution can be that investigation like, say, whole exome sequencing, which utilizes techniques to study all 20,000 genes or 22,000 genes together at a time, can be such a broad investigation that it can give us a diagnosis in each and every case. But that is never so. So no single investigation in any branch of medicine can give you all the diagnosis of all the disorders. So here we must understand that we have to choose the investigation, select the investigation, so that is appropriate to the situation and that is appropriate for your patients. So way back when I started my residency, we were doing for serious patients like hemoglobin and hemogram, and then came the CRP. Now the BioFire is there for pediatrics patient, for ICU patient, for rapid infectious molecular diagnosis. But still the place of hemoglobin or hemogram is still there. So similarly in genetics, when I started, it was karyotyping or FISH technique came a little afterwards, or RFLPs were there initially, or Western blotting was there in practice that time in a big way. So these techniques are still stand and they still hold true for some of the patients. So that is why old investigations also do not lose their place in practice. So we must know about everything, which investigations are available, and we must understand the principles behind it, so that which investigation is actually appropriate for our patient, that we must decide. So every investigation has the strength and limitation that we must understand. So a costly investigation may not give you a diagnosis, though it is costly and dealing with so many genes. It may have certain limitations. We'll go through about specific investigations later in the presentation. Older investigations, as I said, they still retain their practice for certain situations. Secondly, there are so many genetics cases, so many genetic disorders, so many are rare things that you may be seeing for the first time. And so many tests are there. So primary consultation or discussion with a geneticist is highly recommended for you to choose that particular test. So that, because these are all long TAT tests, that is turnaround time may be something ranging from, say, to a fortnight to even 2 months or something like that. These are costly tests. So we don't want to choose the wrong test, always spend more time on it and ask the patient to utilize the precious resources that the patient has. So that is why a prior consultation will help you to choose the test. All available information should be shared with the geneticist. So, for example, when you're studying, say, whole exome sequencing, as I said, we are taking the stock of almost 20,000 genes, but your patient is likely to have only one condition. So if you are able to give full information regarding the clinical situation, clinical findings in that patient, the investigation that have been so far, then the bioinformatician, the analyst will narrow down to a particular set of genes. Now there may be 10 genes, 20 genes, depending on the condition. And then even a single change, which has not been significant, which is a very novel one, which has not been reported before, will be highlighted and will be studied in detail. So that is why from a doctor who is the primary care provider, we expect to get all the information. So it is mandatory. So it is not a blanket, or you're not doing, say, sort of study regarding the efficiency of that test. So you have to give information and then it will become fruitful exercise. Clinical diagnosis sometimes is very evident. The diagnosis is written on the face of the patient, like Down syndrome. Still, we want to do karyotyping for Down syndrome. There are types of Down syndrome. Similarly, sometimes we reach a diagnosis by some indirect techniques for a particular disorder. For example, I'll give like Duchenne muscular dystrophy, where the clinical symptoms with Gower sign, muscle weakness, calf hypertrophy make you diagnose the condition clinically very easily. Or say, take example of thalassemia. So thalassemia, what we do, a child which is becoming rapidly anemic, say beyond the age of say 6 months or so, has hepatosplenomegaly. Then for Duchenne muscular dystrophy, what we do is we do CPK. So that CPK, when it is raised in 1,000, we say that our diagnosis is clean, DMD is there, Duchenne muscular dystrophy is there. Or for thalassemia, which is anemia hepatosplenomegaly, which is progressively increasing, we measure hemoglobin F. And then we say this is child has thalassemia, it has got hemoglobin F of 80%. Now remember, though this diagnosis is clean for your patient, still this is not the genetic diagnosis. Why this is not the genetic diagnosis? Because this CPK and your HbF are actually side phenomena for that particular patient. So actually the gene which is affected in Duchenne muscular dystrophy is dystrophin. And the dystrophin levels are low in muscles, but we never measure dystrophin, but a side phenomenon which makes the cell membrane weak, more porous for the CPK, which is usually present muscle and go to the plasma, that we measure that. And then we say, because it is less than 1,000 with other clinical symptoms, that the patient has Duchenne muscular dystrophy. But still the mutation, the change that is the genetic change that is responsible for that particular patient is totally different. And this matters for us because for prenatal diagnosis, we want to actually have a look at this particular mutation. We want to look for that in the prenatal sample, either by chorion villus biopsy or amniocentesis. So that is why we must know regarding this. So though you have the clinical diagnosis. Similarly, for thalassemia, there are 250 mutations that are responsible for beta thalassemia. So which of these mutations is the cause of thalassemia in your patient? This will understand by studying the DNA of the patient, not by having a HbF examination. So whenever we reach a diagnosis by this common means, these are cheap means, very easily available. Still remember that it's not the genetic diagnosis. And getting a genetic diagnosis is important. It is duty of every primary care provider. And lastly, the cascade screening. Why cascade screening? Cascade screening means that you trace the first order relatives one by one and then screen them for the carrier status so that you will be able to have a prenatal diagnosis in those when required in time. So which are the investigations that are available in genetics that we will see in today's lecture? What is the basic principle behind each one so that you can be making your decision more appropriately to select correct investigation? And we will also have a look at the common lab reports that are from genetics, because the lab reports are quite complicated. So I'll just try to make them a little easier for you to go through. So genetic disorders have different types of inheritance. So this all of us know. It's our human being has a body made up of cells which have got 23 pairs of chromosomes, out of which 2 are sex chromosomes, 22 are autosomal pairs. On this 22, these are the one, of course, half the set is coming from mother, half the set is coming from father, and there are 25,000 genes. So on each chromosome, there are around 20,000 genes, 1,000 genes. So whenever you are studying chromosomes, you are not going to the gene level. So gene level the diagnosis is required for single gene disorders. Single gene where a single gene is affected. Similarly, there can be other defects like say common or birth defects where multifactorial inheritance with external factors and interplay with say 3-4 genes is there. For example, our open neural tube. This is polygenic multifactorial inheritance. There can be some novel non-Mendelian inheritance also, which do not behave according to the laws laid down by Gregor Mendel, the pioneer geneticist, you can say, as they stand for the single gene disorders. And obviously mitochondrial disorders, because mitochondria have got their own circular DNA. Now, when you come to the chromosomal inheritance, the chromosomal disorders, we can classify them into disorders of chromosome number, like Down syndrome or trisomy 18 or Edwards syndrome, Patau-Turner, Turner is monosomy X, or structural aberrations like, say, single chromosome may have some deletion or duplication can be there of certain part of chromosome, ring chromosome, translocation, there's some part of chromosome goes and it relocates onto some other part. And so there's this other entity which is known as microdeletion syndrome. What is microdeletion syndrome? These are the syndromes which occur because of a segment either duplicated or deleted, which is not made through the microscope. Now, actually, chromosome karyotyping, when we do perform karyotyping, is the visible structure of chromosome that we see through microscope. Is the photograph that we take and then arrange the chromosomes according to their number, according to their type. So that is the karyotyping. But this visibility is limited by the resolution of the microscope. So whenever a deletion is less than, say, 5 MB, it cannot be made out through the microscope, and then it becomes a microdeletion. So we'll see what examples of this a little later. And then obviously the Mendelian disorders or single gene disorders, two of which we just now talked about, where Duchenne muscular dystrophy, which is X-linked recessive, or autosomal recessive disorder like thalassemia. So there are different types of DNA changes can be there, which are responsible for that particular disorders. They may be point mutations, or what it is presently known as SNP, single nucleotide changes. Or there can be deletions or duplications, where you can say that multiples of nucleotides can be either deleted or duplicated, which is more common, say, with a condition like Duchenne muscular dystrophy, or the famous condition of Huntington's chorea or Huntington's disease, or even more common than that, the Fragile X, are because of triplet repeats of certain orders of the combination of triplet codons. So these are go on repeating, and when they increasing number, they cause the condition like Fragile X, which is the second most common cause of mental retardation after Down syndrome, which is a chromosomal cause. So I will just now talk regarding the what tools are available in our this genetic odyssey, diagnostic odyssey. And purposefully, I have taken these first two tools because we can never forget them, we can never replace them. And that is our history taking, our pedigree drawing, and examination. This is very important because, as I said, it is going to give us clues regarding the differential diagnosis. Secondly, the routine investigations like our hemogram, our HPLC studies, as well as our other studies, X-rays, CT scans, MRIs, these are routine, easily available investigation everywhere. They are also going to give you help in differential diagnosis as well as diagnosis. And this is important for each and every case. I will illustrate this by giving one example from my own practice where I missed a diagnosis initially. So as you can see here, this baby is around 7 months old, and the father was living at that time was his Middle East, and the baby was living with mother and at the mother's parents' place. And this baby was brought to me for little large skull. As you can note, that baby has a dull look. Baby had excessive crying also. And then I was not getting the diagnosis because I had not looked at the child closely. I was not considering any condition that way. And then picture became clear when this paternal grandmother visited the place, and she came to my clinic with the child, and you can easily see here the diagnosis, that is neurofibromatosis. Then I looked at the child closely, and what you can see on this child's back are these multiple cafe au lait spots, not this mongoloid spot, but this you can see the small spots. So these are multiple in number. So this is a harbinger of neurofibromatosis, which may can later take, say, this particular full-blown condition. But initially in a child, it may manifest with, say, multiple cafe au lait spots with little large skull. In 10% of them, some delayed milestone mentally subnormality can be there. So this was the diagnosis. I missed it because I did not look at the child close in detail. So every patient that you get, please examine thoroughly, note down your findings, have a pedigree drawing. Had I asked regarding the pedigree, then I would have known regarding this case. The father was also affected, but little mildly than this mother. So history and clinical examination provides you diagnosis in many instances, I would say majority of instances. And also it helps you to plan investigations. And why planning of investigation is important, you can see here from this particular case. So here you see that this child is about 5 months old when this was brought to the hospital, and that time child was operated for cataracts. So young child had a little bit of jaundice initially and cataracts. So combination was considered to be responsible, so pointer towards galactosemia. And that child was thoroughly 3 times was investigated for galactosemia, but the other things were not looked at. So when the child was brought to me, I was lucky because child was a little older. You can see the broad face and was a little shorter appearing hands, and X-rays revealed that the long bones had metaphyseal dysplasia. So child also had hepatosplenomegaly. Now, all this combination is indicative of chondrodysplasia punctata. You can also see a little bit of punctate calcification in the knees. Rare condition, rare diagnosis, but clinical examination is giving you the clues regarding the underlying diagnosis. So the actual investigation needed for this particular child was that should be have to be there regarding the peroxisomal disorders. So history and pedigree examination will never be replaced by any other technique. So that is my message to you. And routine investigation will also be informative. What other things that we have for investigations with us is something like initially were old investigations like karyotyping. That is our chromosomal examination. And other thing is fish. So fish is not our edible, what we eat actually as a delicacy, but fluorescent in situ hybridization. That is one technique which is used to study chromosomes rapidly. Other investigations we have discussed regarding indirect evidence, for example, CPK or hemoglobin F, or evidence of function like say enzyme studies, or something when we want to have a molecular or a single gene diagnosis. What other investigations that we have are MLPA, or that long falls we'll see later, or polymerase chain reaction, a very famous and very old and very well established investigation. Other I have not mentioned, there is the Sanger sequencing, and the new kids on the block presently to study the chromosomes and the microdeletions and other things is a technique which is known as microarray or cytogenetic microarrays. And the most famous one in which everybody is interested is next generation sequencing or exome sequencing. It may be clinical exome sequencing, focused exomes, or a whole exome sequencing. So all these techniques that are available to us, and we have to choose the perfect technique which is suitable for your patient out of these techniques. So as I said, there are different types of inheritance. And whenever we are dealing with disorders of chromosomes, we must know that they are because of either number changes, they can be additionals, like Down syndrome or Edward-Opatow, which is known as trisomies, or monosomy, which is responsible for Turner syndrome, monosomy of the X chromosome. Or they can be disorders of structure, like deletion, duplication. As I said, on every chromosome, there are around 1,000 genes. So a deletion, say, of even a small part can be a deletion regarding, say, 20, 30 genes also. So which can play havoc with the clinical condition of the patient and may be responsible for a serious disorder. And even 20, 30, we cannot make out through microscope. So that is why these are the microdeletion syndromes. So all these are the types of chromosomal disorders only. And the technique, basic technique that we have is to study chromosomes is karyotyping. And we know that karyotyping is done from our peripheral blood cells, and chromosomes are present not in RBCs, but in our white blood cells. But these white blood cells are actually mature cells. So after they become lymphocytes or neutrophils, they don't divide beyond that point. So we stimulate them with phytohemagglutinin and make them divide. Now this is a mitogen. And because this is meant to divide, and this we want to do, because otherwise chromosomes are there in a cell in a entangled form. So at the metaphase stage of the cell division, they are at their most condensed form, and then we can study them, take a photograph, then arrange them. So that is why we want to stimulate them to divide, arrange them at metaphase by adding colchicine, which is a drug that you use for gout. And then the cells are fixed. Then we'll have a look at them, take photographs through a microscope after staining them. And then we arrange them. So all this process takes time and the culture also. So that is why it is a time consuming, very subjective process. Expertise is required in this. And this is a metaphase spread. That's one metaphase, that is one cell's chromosomes are being photographed. at high power, and these are being shown to you. And this will be arranged into a karyotype or systematically. As you can see here, the chromosomes are given number. The largest one is given the first number, and the smallest one is given the 22nd. Actually, 21st is the shortest chromosome, but 22nd is given. And then there are different, you can see dark and light bands are here. So this is what is trypsin gypsa staining. So these bands are typical for each chromosome. So you can know by looking at these bands, by which if a small portion is lost or added or something, you can make out. Also, one portion of chromosome goes and some attaches to some other chromosome, no translocation, that you can make out by this image, and a lot of expertise is required. So, as I said, there is a resolution limit because we are taking this photograph through a microscope, and this limit is 5 MB. 5 MB is 5 megabytes. So it does not detect small changes beyond this particular resolution. Also, one drawback regarding it requires the dividing cells, and it is time-consuming, subject to expertise required, as I said. But importantly, the strength of this particular investigation is that it is cheap, and second thing is that it is available at many centers, and it takes photographs of actual genetic structures, that is chromosomes. So the things that you see here are visible with the aid of the microscope, of course, to our naked eye, that we can see them. Therefore, this is the investigation which is of choice to make out a balanced structural malformation. Balance means what? The total DNA content of that particular patient will be same. There is no, but there is structural rearrangement. So if supposing a portion of this particular chromosome goes and attaches to some other portion, then this can be seen only and only through a karyotype and not by a CMA. So that is why balanced things, structural rearrangements can be made out by means of a karyotype and not by CMA. This you must understand, though it has got a, as I said, resolution limit and time limit also. So for our rapid diagnosis, the technique that came into being after this karyotyping was, as I said, fluorescent in situ hybridization. Now, in all the genetic techniques, what is made use of, this is property of DNA, which is known as hybridization. So where we know that our DNA, in nuclear DNA, is double-stranded, and the complementary DNA is there on the other strand. So this they hybridize with each other. So nucleotides hybridize with each other, and this particular property is made use of in each and every technique, right from PCR to this fish, even to our NGS, and even to our microarrays. So there, the complementary sequences of DNA go and bind with each other. So in these fish techniques, we have certain probes. And what are these probes? They are fluorescently tagged sequences of DNA. For example, if I have a probe which is specific for a chromosome, then it will go and attach to that particular chromosome only. So here I am showing you fish for rapid diagnosis of aneuploidy. So rapid means what? This is a cell only, and there I have 3 probes which are going and attaching to the particular chromosomes only. So there's chromosome specific probes, and you can see here 3 signals which are normal for that cell. This is a diploid signal. So one green signal is there, one red signal is there, one blue signal is there. And this does not require the dividing cells. So it can use any cell for this particular hybridization. And unlike the karyotype, which requires almost 7 days for reporting, this can be done within, say, 24 to 48 hours. A report is available. So it is rapid diagnosis, and dividing cells are not required. So this is the advantage of fish. You can also use this for microdeletion syndromes. Now here I am just going to tell you regarding how to read a particular report. So for example, whenever a karyotype report is there, you can see the interpretation of this report. So first number is saying you that the number of chromosomes is 47. Then the second is saying to you what is the sex chromosome complement. And then it is telling you what is the abnormality. So here the total number of chromosomes, as you can see, is 47. Karyotype is XY, so this is a boy. And you can see here that the extra chromosome, that is plus 20, belongs to the 21st chromosomes. And they're all separate from each other. So this is a Down syndrome, the famous condition. And this is what is known as a free trisomy. So why free trisomy? Trisomy because there are 3 Down syndrome copies. as well as it's free because they're not attached with each other. So this is called as free trisomy. Sorry. And then there is other type of Down syndrome. So where we call it a derivative chromosome because this is not normal chromosome. And why this derivative chromosome is formed is because one of the 21st chromosomes has gone and attached to this particular 7th chromosome, 17th chromosome, 14th chromosome. So that is why this is a 14th chromosome with a attached 21st chromosome. Total number of chromosomes is 46 only, if you count them. But this is an abnormal chromosome and which is formed by this joining, and this is a translocation. So this particular type of translocation is known as a Robertsonian translocation, which occurs between two groups of chromosomes. That is the D group, which is 13, 14, and 15, and the G group, which is 24th and 21st and 22nd. So this is the Robertsonian translocation. You can see here the word Robertsonian translocation. These chromosomes are acrocentric chromosomes. Acrocentric means once the centromere of the chromosome goes to one end of the chromosome. The short arms, which are known as the P arms, are extremely short. And unfortunately, I cannot teach you regarding chromosomes only because we are going to talk about all the techniques in a very short time of 1 hour. So this is the karyotype report for a translocation Down syndrome. Now, supposing the number is 45 only, and a copy number of this 21st is the usual the same, then it's a translocation carrier. It's not a Down syndrome translocation, but it's a translocation carrier because the total number of chromosomes is same. Total number of chromosomal material is same. So it is balanced translocation carrier, Robertsonian type. Now here we see one of the Turner syndrome cases with a little milder phenotype. And the abnormality here is that you can see here all other chromosomes are normal. The total number of chromosomes is 46. There are 2 X chromosomes here. So you can see one X chromosome, this X chromosome is normal, and the other X chromosome is showing something different. So as we know that there are 2 arms of chromosomes are called as P arms and Q arms. And here, this normal chromosome has the short P arms and the long arms that are Q arms. But the other, this chromosome you can see here, though it's an X chromosome, the Q arms are joined together. So this particular chromosome is a mirror image. If you have a mirror here, it is a mirror image of Q arms. So that is why iso means identical, and it is a Q arm which is identical. The whole of the Q arm is sort of duplicated and P arm is absent here. So this is a type of milder Down syndrome manifestations can be there, and this is a isochromosome X. So here now there is a different translocation where you can see that one of the, you can see here it is a translocation between 6 and 13 and the chromosome 1 also. It's a complex translocation. So there are 3 chromosomes taking part in this translocation. So it's a very complex rearrangement that has happened in this particular karyotype. And these 3 chromosomes are lost or gained, and they've gotten relocated or translocated into each other's arms. And this is a karyotype containing 46 chromosomes in total, but this have got haywire. So this is a very complex rearrangement. So genetic counseling is definitely recommended for this. Now we are going to the next investigations, which is there in practice. And I must remind you that this is there in your day-to-day practice. Because this is not a research tool anymore. So it is easily available with many labs. And this is used to make out even smaller imbalances, which cannot be made by through a microscope. This can be made through this technique, which is known as CMA or cytogenetic microarray. And there are different types of microarrays. We'll not go into those details because basically many of these microarrays utilize a combination of these probes. So that is why just remember microarray. So microarray detects all imbalances which are detectable by karyotyping. So microarray can detect the aneuploid, the mistake regarding the number of chromosomes. It can also detect microdeletion or microduplication, which cannot be detected by a simple karyotype. And the resolution, as you can see, the resolution of a microarray, a simple microarray can be something like say 100 kb for a, and a very high resolution microarray can have a resolution of 50 kilobytes. So you can see that it's almost 1,000 times as powerful or more powerful than a karyotype. Make out the small changes, but it can make out only the imbalances. It cannot actually make out the balanced ones. So for imbalances, you can say that microarray is the test of choice. But if you want to make out, say, karyotype, say balanced structural rearrangements, then karyotype is the test of choice. Another advantage for microarray is that you can directly perform into the any tissue. So a culture is not required. So it is that way, culture is not required. Is an advantage, especially for some tissues where you cannot grow at culture. But at the same time, we must remember that DNA quality has to be good. So that is why that is always tested before, and if that is good, only then this investigation is proceeded with. Now CMA, as I told you, it has a good diagnostic yield as compared to the microarray, and it's much better also. So that is why you can see the second reference that the CMA in conditions like developmental delay or intellectual disability or autism spectrum disorder, 12% better yield than conventional G bending that what we have just now seen. So that's why American College of Medical Genetics, which is the sort of body which gives us very useful guidelines, has recommended that CMA should be the first year test in non-syndromic. Non-syndromic means what? You cannot know by looking at the patient with which condition is there. Otherwise, you will use only that particular investigation. So non-syndromic developmental delay or autism spectrum disorder. So ACMTMG guidelines presently are telling us this. I'm sure that they are going to change it in a few months' time. That CMA testing should be the first line test for all these conditions. Multiple abnormalities where no specific syndrome can be is not accounting for that, or apparently no clue is there, non-syndromic, no external features are there, autism spectrum disorders also. This is the ideal investigation. But of course, whatever findings we must get through this, we must follow them up with appropriate follow-up techniques in either SIPs or parents of that particular patient. Now, this is how a CMA report looks like. Now, we'll see here what was detected. Now, this is not the actual image, but these are the ideograms of the chromosomes. So this is not an image taken through microscope. So this is how a report of a CMA will look like. The concerned chromosome is the 15th chromosome. What is deleted if this particular region is deleted on the Q arm? The number of genes is 20. So that also the CMA will tell you which genes are likely to be there. But now there are various databases that a bioinformatics or medical geneticists uses, and they tell us regarding the significance of this particular change. So here you can see here that we are not sure regarding the clinical significance because there have not been previously patients reported. This is not absolutely normal also. So that is why whenever such change is there, then we go back to the databases or previous literature to check the significance. Also, one more thing that we check in exome reports as well as CMA reports is whether the change that we have with us matches the phenotype, that is the clinical features in this particular syndrome. So this is a complex exercise, and always you will see that a line mentions that please correlate clinically and genetic counseling is indicated. Each and every report will have that because each and every situation is different, each and every condition is different. So though we may have a diagnosis, still we must have this exercise. So that is why it is written in each and every report. So now we have got 3 sets of investigations with us. One we discussed was karyotype and FISH, and the chromosomes which are actually image. And the size that we make out was the resolution is more than 5 MB, only those changes will be able to detect. A microarray, which is again required for deletion duplications. It makes out changes which are 100 KB or very high resolution, we have 50 KB also or less. That can be made out through a microarray. And then we have what is known as a very good investigation, and that is sequencing. Now, in case of sequencing, the conventional sequencing is not called first generation sequencing, but it is called the Sanger sequencing. Sanger was the pioneer of sequencing both proteins as well as the DNA. So he was again a great scientist, and he he invented both these methods. So that the conventional sequencing, which is nucleotide to nucleotide sequencing, which you can compare with, say, proofreading of our text, can be it can be compared with that, is the conventional sequencing. And what we have presently is parallel sequencing of multiple genes which go on simultaneously. Then it is called next generation sequencing. You can say this is our generation sequencing. So it is the next generation. I'll see why it is called so and what is the procedure very shortly later on, where to apply it. But important thing to know here is 100 kb, and here with this sequencing, what you can make out a single base pair to even 1,000 kilobases you can make out with the help of sequencing. And various algorithms are presently now there, which can tell us, and probably next few days, in recent future, maybe sequencing can replace microarray also. So that is possible. Sequencing getting more powerful and more very detailed algorithms are now being built in, so that sequencing can take place of microarray. And not well into the type of topic which I like very much. So that is our sequencing. Now let us see how we apply all these techniques that are there with us to the patient. So if we have a patient and the diagnosis is very obvious to us, like a Down syndrome child, we obviously should have a history examination to assess the status of the child, how to support him. So our treatment is with either occupational therapy or your speech therapy, whatever, physiotherapy when required, endocrine support if required. Then you anticipate certain things like, say, cataracts and other things. And then when you're supporting this, you also want to know regarding the karyotype, that is the chromosomal pattern which is responsible for his condition. Though the diagnosis is very obvious, we must have a karyotype. And if a fast diagnosis is required, we can have fish, especially in prenatal situations, that is on CBS or our amniocentesis. So why this is particularly required to karyotype is because, as I said, free trisomy, that is trisomy 20, where all chromosomes separate, has got only 1% risk of recurrence, or it is age-appropriate risk of recurrence only. While if the translocation is there in the Down syndrome, and one of the parents has a translocation, is a translocation carrier, then depending on the risk of recurrence, the translocation and which parents it originates for, the risk of recurrence will vary. So for a 21 translocation where two chromosomes are joined together 21st, then the risk of recurrence is 100%. So the progeny for that particular parent either is going to be a Down syndrome or first trimester loss. So therefore, for this particular, we have to replace that gamete if they want to have a baby. They cannot have biological baby. So that is why we must know regarding the karyotype of the Down syndrome, then trace the parents, culture them, and then know the type of translocation, which of the parent is the carrier. Then Williams syndrome. So Williams syndrome I particularly took because it's quite a common syndrome. And it's because of small deletion on the Q arm of the 7th chromosome. The gene important here is the elastin gene. So this particular child, as you can see, has got full lips. Then the child has also got this particular dysmorphism. The eyebrows, you can see the medial flare of eyebrows is there. And this dysmorphism, along with the heart region, has made me suspected the diagnosis of Williams syndrome. And the method for diagnosis will not be the karyotype. What I want to have here is fish for that particular region. The probe will be there available, and then I can have a diagnosis of Down syndrome. As I said, for developmental delay or intellectual disability, autism spectrum disorders, how should we diagnose them? We should diagnose them by having a microarray. So that is the appropriate investigation, and of course, all of the investigation. Yes. Lecture on it. So these are all these ones. So MLP, I will talk about this technique little later, can also be tried wherever fish is available. So why that is, so I'll just tell you. So let us go to the molecular studies and what are the techniques that are available to us. So this we are usually apply for single gene disorders, or what we popularly also call them as Mendelian disorders. And these Mendelian disorders, you use either a polymerase chain reaction or Sanger sequencing or next generation sequencing, that is exome sequencing. Mansi? Hello? I think
Yes, sir.
I may go beyond this because I require at least 25 minutes after this. Will it be okay?
Yes, I think it is.
Yeah. So polymerase chain reaction is a reaction, as you can say, it's a chain reaction. And what it is required here is the polymerase, that is DNA polymerase is the enzyme used. And this forms polymers of a particular sequence of DNA. And this sequence of DNA is decided by the probes, that is the oligonucleotide probes that we have, which go and attach to a particular piece of the DNA, which is denatured. So you can say that this, whenever we have this, our double-stranded DNA or the patient's DNA, we heat it so that it gets separated, and particular primers, these are oligonucleotide primers, they will go and attach to those particular primer interested areas, and between them, they will have a polymerase, DNA polymerase acting, and this reaction will occur many times using the DNTPs that we provide, and the specific piece of DNA, specific segment of DNA will be multiplied so many times that is just like a chain reaction of our atom bomb. So we'll see here that the first 2 copies are formed in first cycle, then it will form the next, these 2 copies will form the next 2 copies, that is the 4 copies. So at the end of 35th cycle, something like say 16 billion copies will be formed. So this is what is known as a polymerase chain reaction. And multiple copies are formed. These were initially required because initial techniques used to have something like say autoradiography or electrophoresis, or so that something like that was required. So multiple copies were required. And then you can get this particular piece of DNA, and conventional sequencing, you can employ it to it or measure this particular. You can also strategize your polymerase chain reaction in such a way where it cannot, if a mutation is there, then a different chain will be produced or not produced, or something like you can just use the sort of a combination of primers, like that arms reaction, as we call it, we use that. So now there is one more condition. So this is for the single gene disorders. And this is, for example, for thalassemias, a disorder like thalassemia or sickle cell anemia or our hemoglobinopathies, what is required is just simple PCR, especially for that particular portion or for that particular mutation. Also, whatever single nucleotide changes that we get in our exome sequencing, we use either this PCR or Sanger sequencing only to confirm or to trace in families. So that is why this, as I said, though these are old investigations, still they have got their own place in practice and their importance in practice. So one condition, as we said, discussed before, was the triplet repeat disorders. And one of the most common triplet repeat disorders we encounter when we are dealing with intellectual disability or autism spectrum disorders in males is Fragile X syndrome. So here it is just next common incidence as to Down syndrome in 1 in 4,000 males, which is very high incidence. So this is X-linked semi-dominant, and you can see this clinical picture here. They got prominent jaw, then prominent ears are there. Whenever they get into puberty, they have a very large size testes. Behavioral abnormalities are there, and the longish face, you can make out this longish face in this boy here. Hyperextensible joints can be there, and females may have milder features. Some of the carrier females may have ataxia, or they may also have something which is known as premature ovarian failure. So premature menopause can be there. So history can be there, where mental uncles are affected with, say, some mental problem, mental abnormality, or and then the boy has got a speech delay. It's a classical history in case of Fragile X syndrome, where you don't use exome sequencing, or a simple PCR, but what you have to order is something like which is known as a triple prime PCR. Now, this is a different technique which is going to give you the count of triplet repeats. So that is why it is called as triple prime PCR for mFEVR1 gene, which is the gene involved in Fragile X syndrome. Now, the report will come to you something like this. So where for this particular child, the number of repeats was 206. So you'll see here that for it is more than 200. So which is whenever it is more, then the diagnosis is clinched. Also, something is known as pre-mutation. So in case of pre-mutation, the manifestations may not be there. But that particular, in a female especially, she can be a carrier and may have certain problems later in life. So full mutations is present in males and is being the X-linked disorders, mental retardation is common is males. But some of the females, if they got a full mutation, they can also have mental subnormality. So it is an investigation which is to be used judiciously and should be the first investigation in case of a male who is not a Down syndrome but has mental subnormality or autism spectrum disorders without any other clue. Now we'll go to this particular technique, which is popularly known as MLPA. Now I suggest that anybody who has this the short form is popular, just know and try to understand what this stands for. Because when you understand what this stands for, this will give you whole technique in a nutshell. So MLPA means multiplex, many reactions will be occurring, ligation dependent. So this is something different, and it is probe amplification. So here again, the probe amplification means something like, so PCR is only used here. So here what is used is that a long gene side dystrophin is present. Then we have, so dystrophin is, as I said, it is responsible for Duchenne muscular dystrophy. And it has got, the gene dystrophin has got 79 exons. And these exons are, when the deletion and duplications, it is responsible for almost 85% cases of Duchenne muscular dystrophy. So that's why we want to go in not for Sanger sequencing, because it's a very long gene. We want to go in for the deletion duplication studies by MLPA. And this custom-made these probes are there. We specifically go and bind on to the 17 exon, depending on the sequence of those exons. And then they have what is known as the, now these particular probes first initially go and bind, and then this ligase is provided. So wherever they bind, this ligase is acts. Whenever they don't bind for that particular DNA segment, this ligase will not act. And then a reaction is carried out for multiplication of these particular sequences, and whichever have attached will go ahead with the PCR reaction. Whichever are not attached, they will not produce the reaction. So in this way, then the analyst reads this and then knows regarding the signals and whether a particular segment of dystrophin gene has deleted or duplicated is made out through this multiple ligation dependent probe amplification. That is MLPA for you. So as I said, this particular reaction is used for two very common muscle conditions, namely Duchenne muscular dystrophy, and the other one is spinal muscular atrophy, where deletion of exon 7 and exon 8 of SMN1 gene is the responsible cause for almost 95% cases of spinal muscular atrophy. Now, there one other reaction that is available is multiplex PCR for DMD, that is Duchenne muscular dystrophy, where 20 or around that probes are used. But presently, because it deals only a small number of mutations, that reaction is particular technique is not anymore used in practice presently. Then we go on to Sanger sequencing. As I said, it is just like conventional proofreading. So it uses DNA-dependent polymerase to make a complementary copy of a single-stranded DNA template. And one by one, this particular nucleotide is read. We'll not go into details of technique for want of time. It's not required also for you to order that investigation. But what is required for you to understand is that it's sequencing both exons and introns. We're not going to the gene structure, but all of us know that our genes are only 1% of our total DNA. And each of our genes, or most of our genes, are formed by two types of DNA, one which express or translates into message, And that particular portion, because it is expressing, they're called as exons. And other portion is called as intervening sequences. Because they're in between the exons, they're called as intervening sequencings. And these are the IVSs. So these are called as introns. So Sanger sequencing, because it goes by nucleotide, not only it reads the letters, as I say, in the manuscript or on the page, but it also reads the spaces in the page, which are the intervening sequences or the DNA sequences that are there. So that is your Sanger sequencing or conventional sequencing. Now, I must say here that Sanger sequencing is still the gold standard for DNA sequencing. So whatever changes you get, even in whole genome sequencing or your exome sequencing, for them to validate that, we must have for that particular region a conventional Sanger sequencing. Once that Sanger sequencing report is there, that this particular change is there, then we take that report as valid. It is effective, very effective, and in practice for sequencing small genes because it is quite cheap that way. But whenever we want to go in for sequencing of a big gene like dystrophin or something like, say, our NF1 gene, which is neurofibromatosis gene, then sometimes we resort to our exome sequencing technique. And whenever we want to have a look at a particular group of genes which cause a similar phenotype. For example, epidermolysis bullosa is a group of disorders which has multiple genes which is responsible for that. So we want to study one set of genes which can produce the same phenotype, then what we use is exome sequencing. But for a single gene, which is of a conventional or say limited size, where it can be practical, we use Sanger sequencing, as well as to validate the changes that we find in exome sequencing, we use Sanger sequencing. Sanger sequencing, again, we have to have a probes for that particular sequence. So this cannot be studied, and on this genetic disease we cannot study, but we can have novel sequences found out through this Sanger sequencing that is there. But unknown disease we cannot study with this. Diseases where multiple genes, as I said, to your set of genes which may responsible for a similar phenotype, genes with multiple exons, I just now named few of them, and it becomes very long for all this particular combination. So wherever we have a condition which is very common, like thalassemia, where multiple variants can be there, then we can just look at which variants are very common. So for our state of Maharashtra, maybe 80-85% of the thalassemias belong to only 5 mutations. So there we can study these 5 mutations, then these reactions can be cheap also, and then use only PCR. But it's a small gene, just around few nucleotides are there. So conventional Sanger sequencing is also practical because it is not that costly as other genes. So 3 things that we must do in thalassemia is that even if your diagnosis is ready with your HbF and clinical picture, still try to study this molecular diagnosis because that will give you a backbone for your prenatal diagnosis. The background also will be there. You can target the prenatal diagnosis within time. Advice regarding iron ore is must whenever you have diagnosis. Supplement the baby with folate and B12 because it's hemolytic set. And index get hemolysis, mutation analysis will give timely prenatal diagnosis. Also, don't forget that there is 50% chance that the first order relatives will be carriers. So that is father's brothers or mother's brothers. Many of them will be in that childbearing age group. So please try to have a carrier status reading for them, and then you can offer them genetic counseling if they require prenatal diagnosis. So your counseling is not finished till cascade screening is there. Now here you see 2 children, unfortunately 2 brothers, and they're having oversight. So both of them were affected by Duchenne muscular dystrophy. Calf hypertrophy was there, CPK was raised. So here, as I said, whenever an index child is, so for this older boy or elder brother, if we had a prenatal diagnosis ready, then we might have had a prenatal diagnosis for the younger one. We could have prevented the birth. So history examination, molecular diagnosis should always be up there. And also the carrier diagnosis in female relatives should be the first order female relatives. So that's why it is important to study the Duchenne muscular dystrophy, as we have discussed. all these deletion duplications are responsible for almost 85% of situations. The rest of them can be either point mutation or deep intronic changes, for where you can employ your dystrophin sequencing or NGS, try NGS first. Now, this has also become important because maybe in a few days, some gene therapy will be available, and for a patient to get a gene therapy, we must know what the mutation is and whether that gene therapy can be applicable to the patient. So that's why we must have a molecular study for that particular patient. So as I said, limitations for Sanger sequencing is patient with unknown disease, patient suspecting with very large gene defect. For example, again, Marfan is just like NF1 and dystrophy is one more condition. A phenotype then can be caused by multiple or several genes like cardiomyopathy or epidermolysis bullosa or your muscle disorders. And then the exciting thing, everybody wants to listen to this particular thing only, that is next generation sequencing. Now, other name for this particular condition is massive parallel sequencing. Now, it is called massive because all the genome or all the exomes is being sequenced simultaneously. So that is why it is parallelly, all the genes are being sequenced. So like I said, we do proofreading of single page, conventional proofreading of a single sentence on a single page. But in case of NGS, it is, if you have got a facility of reading, say, our Nelson's 2000 pages proofreading at one time, then that is something which is being done in next generation sequencing. Or something like if we have a focus. So at our home, we make one dosa on one pan. But when we go to a hotel or when a chef you see at a 5-star hotel, then on a very large pan, that chef is making, say, 20 dosas at a time. So parallelly, he's making 20 dosas at a time. So that is why your next generation sequencing is something like that only. Now here what happens is that your double-stranded DNA is broken down into many copies according to the sequence size or the number of genes that you want to study. And there, these copies then are aligned, and then you sequence them simultaneously, parallelly. So that is why this is known as massive parallel sequencing also. There are different techniques, or will not go into details. But important thing to remember here is that there are 3 different types of next generation sequencing, or 3 different groups. One is which is known as a clinical exome sequencing. So in case of clinical exome sequencing, these are around, say, 6,000 genes where important disorders which can cause rare disease, known disorders, they are taken up. That is our OMIM database. Sometimes it is called as OMIM sequencing also. So OMIM is Online Mendelian Inheritance in Man, and this database is freely available on the net, where it was set up by Victor McCusick, a very famous researcher at Johns Hopkins, and there this particular database is maintained. So any condition, single disorder, single gene condition, you can go there and just have a look at it. So this particular set of, say, next generation sequencing uses probes for these particular things only. So maybe 6,000, 7,000 genes are sequenced at one go, and whole gene is not sequenced here. But what is sequenced here is the coding part. That is the exon sequencing. So that is why it is clinical. Clinical because clinical conditions, important clinical conditions when sequenced here. Exome is sequenced here, and sequencing obviously. And then the second is the whole exome sequencing. So previously whole exome sequencing started with, say, something like, say, 14,000, 15,000 genes. Then they become 18,000. So presently many labs are adding new genes to this. They take up important genes, and there they take up almost, say, presently 22,000 genes are being sequenced simultaneously. So wherever you have something which is evidently genetic, but you are not cannot be focused, say, narrow down to something very specific, then you use whole exome sequencing. As I said, algorithms are now being built into this so that we can have a diagnosis of multiple conditions at the same time, along with a, say, deletion, duplication, or CNV, that is copy number variant decoding capability. So microarrays, they will be easily able to replace in coming days. And the whole genome sequencing. So whole genome sequencing is you sequence everything that is available to you. You sequence everything that is the whole genome you sequence. Now, this is a very, you can say, extensive exercise. You can also see the amount of data that is being analyzed. So in case of whole genome sequencing, almost our whole hard disk is getting sequenced. So that much amount of data is there. 90 to 100 GB data is there. While case of say our whole exome sequencing is something like a pen drive, and for our whole clinical exome sequencing, it is our old pen drive, our card used to have in our phones. So that particular amount of data is there, and this is analyzed by specialized bioinformatics systems. Specialized softwares are available. Some are paid, some are not paid softwares, and a new breed of bioinformaticians or analysts is there. They, with the help of medical geneticists, they make sense out of all the changes that we get. And why I said that, the three terms that you must know are the read depth. Now, obviously, this barrel sequencing goes on occurring because, as I said, it's a reaction. So there, how many times that sequence is being read gives you the fidelity of that particular change that you are getting. So many times the sequencing read, and if you are getting a change, same change in those reads, that means that the read depth is good. But if it is shallow depth, and you are, for example, if the sequencing is being read only 20 times, and you get some change 2 times, then that may not be the valid change. But if you get you get say 70, 80 times in 80 reads, then that sequence, the read is very good. Second thing is coverage. So coverage is how much portion of the gene is being covered by the probes in that particular exome. So that is also very important. Sometimes you may have some change in some your exome, but that portion may not be covered. So whenever you are suspecting something, but you are not getting what you are getting this one, then you must ask the lab what is the read depth and what was the coverage of that particular gene. So that will give you whether we go any further for that particular condition. The changes that we get are, we know that our codons are triplet codons. And for one amino acid, there can be sometimes with 3 codons. For single amino acid, there can be only 1 codon. So whenever we get something, say that the amino acid change will not be there, but we can get a single point change, then that particular thing will become as a benign variant, or a likely benign, because they are reported in databases with very large frequency. Because now this database is also available, something like say ClinVar is available. So you can always go and check in that what is the significance of this particular variant. Then it is pathogenic, similar type of phenotype, and this change was being there. So for example, if a beta globin or sickle cell the changes, it will definitely be pathogenic because it's already reported. Or likely pathogenic, where in silico parameters tell us that this can be there, or there will be few patients, few instances that it's been reported. So that is likely pathogenic. And the third is the variation of unknown significance. So it is something in between. And here, what we want to have is further studies, either with unaffected sieves or severely affected sieves, or with parents, or with in silico parameters, or maybe sometimes even with our functional studies. Sometimes it may be required if the phenotype is matching. So all this is then goes into research thing if it goes to say functional studies. But rest segregation, this is called the segregation analysis. So follow-up testing for parents and sieves is usually required for something, if a change you get as something as variation of unknown significance. So here you must tell the patient that though this investigation we're carrying out, we're studying so many genes, but to match, if we get some change like this, maybe further studies will be required. So we talked about this, and this is the sort of report that you will get here. So pathogenic variant, positive of the reported phenotype was identified in this particular patient. You can see these two changes were identified again. So this particular report is a very easy report to read, very good report to have, because you don't have any further headaches. Sometimes you may have this particular, because this is a pre-symptomatic screening. No, I think this is not pre-symptomatic. Previous children were affected with different phenotypes, different clinical conditions, and consanguineous marriage was there. So we carried out the, the exome sequencing, whole exome sequencing for the couple. And you can see here 3 conditions with 4 variants are found out here. Now for this particular thing, what we have presently are 4 conditions. So it is very, this couple is unfortunate because many conditions are likely in combination. Risk of recurrence for these 3 conditions separately taking 25%. So it is very unlikely that they may have a normal baby, something like that you have to consider. So just to summarize, we have all these situations with us. So for whenever we have exon level deletion duplication, going for MLTM. Whenever we are suspecting a small target area, small gene, going for Sanger. Whenever we got a combination of disorder, combination of genes responsible for a similar phenotype, then going for either CES, that is clinical exome sequencing, or focused exome sequencing. And if the condition is obviously genetic, then we go in for whole exome sequencing, where we do not have a specific pointer. So all these things we must remember. And WGS is in practice, but it is used rarely. It falls into research domain mainly. Not going to this. So just like to summarize, I will say that it has been said that it is PCR, what was said about PCR, that is one of those inventions like internet. Once you have used it, you cannot quite understand how people managed before it existed. So similarly, I will say here regarding exomes of NGS. So exomes and NGS will be one of those inventions just like internet and PCR. So maybe in coming years, we will just wonder how we existed without them. So just my conclusions and just carry on messages. So history, examination, pedigree, routine workup is the baseline. You cannot escape that. Molecular diagnosis should be achieved wherever possible for us to have a good prenatal diagnostic exercise, good ODC, good advice to give to patients. Cascade screening should be practiced for every common condition, especially like thalassemia and Duchenne muscular dystrophy. When specific disorder is suspected, use the targeted investigation. Special investigation, though costly, though maybe covering a lot of portions of genes still, it may not give you diagnosis each and every case. So test appropriate for your patient should be chosen. For that, pre-test discussion with geneticists is recommended. Down syndrome, do not go in for microarray, go in for karyotype in postnatal situations. For MCMR syndromes, microarray is the test of choice. Expanded newborn screening, we're not talked about this, maybe sometime later if I get opportunity, I'll talk about newborn screening. Genetic testing, but this should be done for all NICU babies. This is the National Neonatology Forum recommendation also. Ultra-rapid exome sequencing. So now exome sequencing has now even reached the ICU setup where we can give reports with a TAT of 5 days also. So this ultra-rapid sequencing, then it becomes just like our bacterial culture, though it is much more productive and it can diagnose rare disease also. So it is practical even for a ICU or PICU setup. So thank you. Thank you for giving me patient hearing for such a long time.
Thank you, sir. It was very insightful and very detailed presentation. So we have some questions. First question is, can we cover CNV, copy number variation, by exome sequencing?
Yeah, that's now we can definitely cover CNVs with our exomes. And presently, as I said, lots of algorithms are being built in. In my lab, we can go even up to say 10 kb also for CNV coverage. So in fact, it is much more powerful than a microarray because microarray has got again a resolution limit. Second thing, what has happened is that exome sequencing has evolved with time, but microarrays have not evolved with time just as much as exome sequencing. So the probes that we use in microarrays remain sort of static. So again, and one more that we have not discussed here, in practice it is required many times, is studying parents as well as baby and the prenatal sample maybe simultaneously. So what we call as the trio sequencing. So trio sequencing has got a much more diagnostical compared to mono sequencing for index patient. So maybe there again, we may have for want of time that is we press for time side prenatal situations, or where we do not have we have got a suspicion of a VUS, we can apply for trio sequencing. So the question was about CNV. So definitely exome sequencing can go in for this one. And in fact, now you should seek for those labs only which give you CNV reports.
So another question I think you have already answered. On what basis is any variant classified into pathogenic, likely pathogenic or VOUS? So I think in your presentation, you already answered this. This was posted earlier.
Right. Basically,
Sir,
these guidelines are freely available at the American College of Medical Genetics website. So there, this how to interpret this, say any change that you get, those are available. So anybody can go and read there. So, but as you said, I already covered in the lecture.
So one more question. They want to know your opinion about NIPT testing on NGS.
NIPT, we don't do NGS on NIPT. So NIPT basically, see, NIPT, the T part is wrong. So we should, first of all, we call it NIPS. So present recommendations and present status does not allow us to call it non-invasive prenatal testing. So it does not stand for an invasive test like amniocentesis and then later on investigations like karyotyping. NIP is non-invasive prenatal screening. So presently the guidelines are there that it is validated and it is available for aneuploidies, that is monosomies and trisomies, for 21st, 18th and 13th, Turner syndrome, and it is also validated for a microdeletion syndrome of 22q minus or Cash22 syndrome. Rest presently what the labs are doing are not presently validated by ACMG. Still, some of them are doing it, and for some conditions, some data will be useful in coming days. At the same time, it is NIPS. So just like our double marker, it should be followed by confirmatory testing. So we cannot take a decision for continuation of pregnancy, or you can take a decision against continuation of pregnancy because it, especially for Down syndrome, the negative predictive accuracy of NIPS is almost 100%. So when it is not negative, screen negative, almost 100% of the times the baby will not have Down syndrome. But in case it is positive, then even in those situations, say in 50% of cases when the risk result, double marker risk result was negative, then 50% chance that the baby may have normal baby, even the screen positive result. In case of high risk result there on double marker, then it's 80% chance that the baby may be having Down syndrome. Still 20% chance is there that the baby may be normal. So that's why any NIPS screen positive result should be followed by invasive testing. That is the present status of recommendations.
So I think we'll take one last question. Other than less time consumption, does NGS technique have any other advantages than Sanger sequencing?
No, what was the first question you said?
So how is NGS other advantages? So other than less time consumption, does NGS technique have any other advantages than Sanger sequencing?
No, NGS, Sanger sequencing, whenever you have particular gene in question, Sanger sequencing is definitely better than next generation sequencing. But limitation of Sanger sequencing is that you have to know the gene first of all. So supposing your patient is there and you don't have a clue whether that particular patient has got this particular syndrome. So then that in that particular situation, exome sequencing is much better than a Sanger sequencing. Now for NF1, if I want to have a Sanger sequencing of nucleotide by nucleotide by nucleotide of all those more than say 70, 80 exons, then the patient will be spending something like say 1,60,000 or something that amount. But if I have NGS for this, then I will be covering almost 85% of mutations of NF1 by exome sequencing only. Rest of the mutations may be either deletion duplications, again for which a particular set of reactions will be available through MLPA. And then only 1% mutations will be there which will not be covered, for which I will have to use a Sanger sequencing. So then it becomes much more practical because there you'll be spending something like say 20,000, 25,000 rupees only. So that's why for a long gene, exome sequencing is much more useful. So Sanger sequencing has got advantages, but NGS is definitely there in practice presently.
So one was, how can we identify if a variant is a novel mutation?
No, novel mutation will give our databases are there, like genome database or 1,000 genome databases, where it will tell you the frequency of that particular variant. So that variant, if it is not at all found, it will say that the frequency is 0%. Then becomes a novel variant. So that is the frequency is found there. And whether your this variant is pathogenic, likely pathogenic or something, that we will know by comparing with the in silico parameters. So we got computer simulations will tell us whether this particular variant is going to affect the protein function, like the say it is in important area, say, or it is in the hotspot, or it is in at some splice site or somewhere, or whether it is going to affect the protein folding or something. And then by that, this particular thing, there is scores are also there. We can predict that. We can also carry out, say, functional experiments with your RNA sequencing. Can carry out that for that particular patient. And then this functional experience will tell us whether this goes to the benign category or this variant can go to the, say, likely pathogenic category. Like that is how we go about a variant which is novel in the databases, which is matching the phenotype, but which is not actually being reported so far.
Thank you, sir. So there is one question about PG diploma in genetics. So I would like to answer that and say that with help of Gambhir sir only, we are in the process of developing a course curriculum and we will be announcing it soon. So we are still in development process. Sir is going to help us in that. So maybe in 2024, it will be available. I would request Center to please conclude today's.
So I profusely thank Professor Gambhir, and I went back to my undergraduate days, and I like his opening statement where it says, everything that is old doesn't mean it is defunct, and always begin with the old methods, the available literature, the lab tests, the basic clinical evaluation. Those cannot be overtaken by the modern techniques. And of course, the importance of genetic counseling, sir brought in. He also mentioned about that clinical diagnosis always can be achieved, but however, that doesn't negate the requirement of genetic testing, and it is something to complete the profile of the patient and for the benefit of academic requirement and scientific as well. That is there. I liked where sir started walking us through pedigree screening and after that how to approach the various tests. And he again said that have the basic investigations and imaging reports in front of you and don't jump into, high-flown tests immediately. I liked that slide of sir's where he gave the various differences between importance of the basic karyotyping, its resolution versus sequencing. That particular methodology, what kind of resolution does it give versus FESH, how much of kbs it can, the deletions below what it can show and above what its capacity is. And then sir thereafter, after talking of all techniques, sir went into certain examples, and in the end sir was very nicely, sir summed it up by saying that it is something that is in a statement which he quoted, which basically tried saying that it is something in days to come or present on date. These methods are gradually becoming inescapable. So it is so very important that we all be educated, informed, and continuously be abreast with these methods. So I thank you all again profusely, all the participants, and we'll continue to engage with Professor Gambhir in days to come. And we'll look for a larger and bigger participation, more and more. Thank you so much, sir.
Thank you, sir, for beautifully summarizing. Even better than me. So thank you, sir.
Thank you all. We close the meeting for today.